is to learn such things, but you learned everything, all these things just from one small module. I hope this is the like helpful MATLAB programming tutorial. Am I right?
Yeah, of course it is.
Yes. And whenever you will be going for coding, this will be worth and very helpful.
Inshallah.
So now it is very clear so
Yeah.
far.
Yeah. Bro, like I'm just asking, like, can you write like what does dx means? Like, that the equation you wrote, can you just
Yes,
explain a bit so it will be easier
exactly.
to and I will comprehend it
Exactly.
later. Yeah.
Yes. So normally in paper we write like this, where, so
Yeah.
that is why I wrote this thing,
Yeah. Thank you.
where Mij is equal to connectivity or adjacency matrix, comma, xi
Excellent. Yeah.
is the current stage, yeah, let's say state of the gene of a specific gene I. Normally we write gene I
Hmm.
at time, a specific time. Okay.
Yeah.
So this is how you also need to write while you are writing manuscripts.
Okay.
Yes. This is very clear now.
Yeah. Can you
Perfect.
also write like a dx and a dt? I know that dx and dt, but can you write it for
Yes.
this moment?
There is no issue.
Yeah.
Dxi upon dt is used here for calculating the temporal dynamics of gene I. ठीक है ये?
Yeah.
कहीं पर कोई कंफ्यूजन तो नहीं है?
नहीं.
नहीं. अब आप ये समझिए, जो कल हमने मैटलैब सिखाया था उसी का आज रिवीजन करके हमने आज पूरा कर दिया, कल वाला प्रैक्टिसिंग भी
Hmm.
आज. ठीक है.
Hmm.
अब आप आ जाइए आगे. यहाँ तक तो कोई दिक्कत नहीं है?
नहीं.
हाँ. अब ये हो गया, जैसे हमने कहा था कि हर रोज मैटलैब कोडिंग भी थोड़ा थोड़ा करवाएंगे. तो अब जैसे अगली क्लास में जब हम पढ़ाएंगे तो उस दिन मेरे फिर वैसे ही शुरू करेंगे बायोइन्फॉर्मेटिक्स एप्लीकेशन से. फिर जब आखिरी टाइम पॉइंट बचेगा तो उसमें हम मैटलैब कोडिंग पे जाएंगे. ठीक है. फिर संडे को जब हम स्टार्ट करेंगे तो फिर संडे को फिर हम ऐसे ही करेंगे कि फिर शुरू में मैटलैब कोडिंग करेंगे, फिर कुछ और पे जाएंगे. ठीक है. ताकि कंटिन्यूटी बना रहेगा आपको. ठीक है ना?
Yeah, bro.
हाँ. So now I am moving to another theme. Okay.
Hmm.
And today's theme is mostly basics of bioinformatics tools. ठीक है.
Yeah.
So now here, basics of bioinformatics. Here what you normally have heard or seen in daily life with yourself or with other people, that there are different types of things. First thing is called, let's say, database. Second thing, web server. Third thing, installation files. Fourth, freely available tools, commercial tools, etc., etc. Okay.
Yeah.
What do you mean by database here?
Where a lots of data is available, like NCBI,
Yes.
Uniport.
Yes. Yes, please go on. NCBI is the database for all kinds of genes, proteins, everything,
Yeah.
okay, related to biological systems or
System.
organisms.
Yeah.
Clear. So this is one of the most commonly used database, that is NCBI.
Yeah.
Okay. Next is EBI.
Yeah.
This is also a database.
Yeah.
Then further, if you go more depth, then it is called ensemble.
Ensemble. Yeah.
Okay. This is also the database. Okay. And similarly, there are multiple databases. Now, when somebody is saying pathway database, then this is also one of the database.
Okay.
Okay. So this is, let's say, I'm saying one thing related to database. Okay. Gene, protein, then KEG database. Other databases are more specific, that is UniProt.
Yeah.
Okay.
Yeah.
Further, what could it be? RCSB. What it is for? Meant for?
Uh, it's for gene, right?
PDB, protein.
PDB protein. Sorry. Yeah. Protein.
Okay.
Yeah.
RCSB.org.
Yeah.
Similarly, there are hundreds of databases these days. Now, somebody can say that TCGA is also a database for cancer.
Okay.
GEO is also a database for everything, all disease.
Okay.
And there are further large number of databases. Now, we are coming for some specific things. Okay.
Yeah.
For example, sequence of proteins or genes. First it is gene, then it is protein.
Protein.
Okay. If I am talking about the sequence of gene, where will you go for fetching the sequence of the genes?
For genes, RC, RC, RCSB probably.
No, that is for protein. You will go to NCBI.
NCBA. Okay.
You will go to EBI. You
Okay.
will go to Ensemble or
Okay.
Uniprot also. But
Okay.
Uniprot is mainly for protein. Okay. So these three databases are for where you can get genes and proteins information also.
Hmm.
Okay.
Hmm.
If you want to get 3D structure or primary structure of protein, you have to go to Uniprot. For 3D structure, you should go to RCSB. If you want the pathway analysis to get information about a specific pathway, you will go to KEG. You will go to TCGA for cancer
Okay.
related data bank. You will go to GEO for all kinds of diseases.
Okay.
So when I'm talking about sequence, then it could be primary sequence, secondary sequence, tertiary, or quaternary.
Okay.
Primary sequence means ATGC, this, something like this, just in one line. Or if we are talking about amino acid, then you know, like glycine, alanine, valine, leucine, isoleucine, something like this. Okay, but you are aware that the primary sequences, the protein or genes, are not like a line. It is always in certain structure, and that structure could be secondary or tertiary. Okay,
Yeah.
and sometimes it also goes to quaternary.
Okay.
In case of protein mainly, tertiary structure is known as functional form of the protein.
Okay.
Am I right?
Okay. Yeah.
Yes. So you always need to either fetch 3D structure of the protein or predict 3D structure of the protein when you are going to analyze the structure of the protein. For this purpose, you need to collect the primary sequence of the protein and then go for 3D structure prediction of the protein.
Yeah.
Now, the protein structures are predicted on several principles. One is called homology modeling.
Yeah.
That means based on similarity.
Yeah.
Or second thing is de novo prediction of protein structure. That means we don't care how the structure should look like, but we predict based on certain software algorithm on our own. But the best structure predicted is always from homology modeling, and mostly the people prefer to have homology based structure.
Okay.
And these days, you might have seen that some people got Nobel Prize this year, and that is on AlphaFold change, AlphaFold 2,
Yeah.
and that is only for 3D structure prediction of the protein.
Okay.
And to predict the homology based modeling, you need to go for alignment. For example, you take the sequence of your protein and go to the protein structure prediction software. They will predict based on the similarity, and similarity is calculated by alignment. Is it clear?
Yeah.
Alignment means what are the sequences portion matching with the other known proteins.
Okay.
So they take the sparse part structures and then they concatenate the structure into one structure. That is what you claim. This is the 3D structure of your protein.
Okay.
Is it clear now?
Yeah. Yeah.
So alignment is for several purpose. One is for mutation prediction also,
Hmm.
sequence similarity also,
Hmm.
clustering also,
Hmm.
and other things are protein structure or DNA 3D structure, RNA 3D structure prediction also. So the last part here, you could see gene protein information, and that you always get from most frequently used software for protein information or gene information. We always prefer to go UniProt these days.
Okay. Okay.
Instead of going to NCBI, UniProt has become more common.
Okay.
What is the reason for UniProt becoming so much popular to use?
I think it's more specified and it gives it's generate like more specific result, right?
Yes. So here the reason is UniProt is frequently updating the database, websites, and frequently integrating the most useful software with it.
Okay.
For example, AlphaFold 2 is now connected with UniProt, not just